So good afternoon. Thank you so much for joining us for the National Sea Grant Law Center's 2022 webinar series. We're so excited to see everyone here today. This is the 5th webinar that we've done this year, and it's been so great to see all the different topics that we've covered. So for those who don't know me, my name is Stephanie Otts. I'm the director of the National Sea Grant Law Center, and today we are featuring work that the University of Mississippi has done related to oyster restoration. And I'm going to let Jessica Pruitt introduce herself, but when I turn it over to her. But we're looking forward to seeing and hearing more about the project that we've been working on together. So just a few housekeeping items. We are recording the webinar for posting on our website after the webinar. So if you have colleagues or others that were interested in attending and couldn't make it, they will be able to watch the recording later. You may put your questions or thoughts in the chat box at any time as we go through the webinar. Right now, we have everyone on mute to cut down on background noise, but we will have time for questions and answers at the end. And so you can either use the chat to ask your questions, or if we have a small enough group at that time, people can come off of mute and ask their questions. And so I think with that, I'm going to stop my screen share and turn it over to Jessica so she can start hers.
All right, so is that the screen up?
Yes, I can see it. I think you're good.
All right, cool. Thanks, Steph, for that introduction. And so yeah, I'm Jessica Pruitt. So I'm a postdoc working on a project with Steph as well as other Embrace funded researchers here at the University of Mississippi. So Embrace is the Mississippi-based Restore Act Center of Excellence. And this is sort of the second iteration of this University of Mississippi team, which I've joined, working on sort of both lab experiments and water quality monitoring in the Mississippi Sound to help inform all the restoration efforts going on there. And so for the next few minutes, I'm going to go over with y'all a policy brief we put together titled Accounting for the Entire Oyster Life Cycle and Restoration Efforts, so which is again based on those experiments and water quality monitoring efforts. So to begin, historically, Mississippi oyster landings have undergone cycles of boom and bust. The Mississippi oyster industry was really at its prime in the early 1900s when Biloxi, Mississippi was known as the seafood capital of the world. So due to the large amounts of shrimp and oysters landed and processed in the city. Yet overharvesting during World War II led to a population crash that recovered under strong management in the 1960s, only suffer again due to a string of natural disasters in the late 1960s and early 1970s. In the early 21st centuries, and Mississippi oyster harvests were actually doing really well. They were averaging about almost 400,000 sacks between 1998 and 2004, but they plummeted after Hurricane Katrina in 2005, in which catches briefly recovered until 2010 in the Deepwater Horizon spill. Also during this time period, there's been 7 Bonnie Carey spillway openings. So this is when they divert fresh water from the Mississippi River into the Lake Pontchartrain that eventually flows into the Mississippi Sound in order to alleviate flooding risk to New Orleans. And so in 2019, it was the most devastating of these openings, where there was actually 2 openings releasing most amount of fresh water from the Mississippi River into the Mississippi Sound. And there was nearly 100% oyster mortality on the public Mississippi oyster reefs. And there's been no public landings since 2019. And so as a result of these declines, Mississippi has put a lot of effort into restoring oyster reef populations. In 2016, Mississippi Governor Phil Bryant created the Governor's Oyster Council on Restoration and Resilience, because the state recognized not just the economic value of oyster reefs, which obviously provides food and jobs for the state, but the ecological values, which are they provide a habitat for other commercially important species such as fish, blue crab, and shrimp, but they provide protection from shoreline erosion and storms. And since oysters feed by filtering the surrounding water to remove algae and other particles that they eat, they also contribute to improved water quality greatly in estuaries. And so the council set an ambitious goal of for Mississippi to produce 1 million sacks of oysters by 2025. And so to make this goal a reality, there's been substantial oyster reef restorations ongoing in Mississippi, with millions of dollars already spent or dedicated for future projects. And these activities have largely involved deploying culture material to areas that historically contained oysters in hopes that wild populations of oyster larvae will settle and over time form a functional reef. And so the rationale behind this is that you'll have adult oysters that are reproducing in the area. So as this oyster life cycle diagram shows, you've got adults releasing gametes that fertilize to form swimming larvae that spend about 2 weeks in the water column until they are ready to settle, or in other words, attach to the bottom, and their favorite surface to attach to is either other adult oysters or oyster shell. And so at that point, they undergo metamorphosis, kind of like a caterpillar turning into a butterfly. Instead, you get an oyster larvae turning into a permanently attached oyster, which we refer to as spat. And the hope is that they will grow into a reproducing adult and contribute to the population. And so the restoration success is really dependent on the survival of these early life stages, but they're generally found to be less tolerant to environmental stressors than adults. But it's really unknown which stressors might be the most impactful and which stages might be the most vulnerable. And you can also have multiple stressors occurring at the same time, creating interactive effects that make it difficult to predict what how oysters will respond. And so things like freshwater flooding events can cause multiple stressors. So either a Bonnie Carey opening or prolonged periods of rainfall. These cause large amounts of fresh water to flow into estuaries that are normally, they're not quite as salty as the ocean, but they're still salty. So this large amount of fresh water is going to reduce salinity levels suddenly and often for extended periods of time. It's also going to bring in increased nutrients, which can lead to formation of algal blooms, which is known as eutrophication. The associated negative water quality issues of hypoxia, acidification, when these algal blooms begin to die and decay, because the bacteria that eats the dying algae also needs oxygen to breathe, and it's often taking a lot more oxygen and taking that away from other animals to use, as well as bacteria respirers too, putting off CO2, which is going to reduce your pH and cause acidification. And so restoration practitioners really require information about how these sensitive early life stages may be threatened by these flooding events that are becoming more common, but which flood-associated stressors cause the most severe outcome and whether they can interact to modify larval and juvenile responses is not well understood. And so that's where our research really focused on, sort of examining the effects of flood-associated stressors on both larval and juvenile eastern oysters to better understand the combined impacts of acidification, hypoxia, and low salinity on early life stages. And so then we translated these findings into the policy brief that I'm discussing with you all today to really highlight the importance of these results for oyster restoration management in Mississippi, as well as other systems subjected to these frequent freshwater flooding events. And so for the science, what we did in the lab was we exposed these 4 different early oyster life stages to either low dissolved oxygen, low pH, or low salinity in isolation or combination, and then assessed the oyster response via growth or survival to these stressors. And so the stressors concentrations or level we chose were actually based on quality conditions we recorded in the Mississippi Sound during the 2019 spillway opening in the first iteration of the Embrace project. And so the next few slides are going to have graphs that look like this one, which get my laser pointer out here. So on the y-axis, you'll have some sort of oyster response. So in this case, larval growth and each of the treatments. So our control treatments, our oyster response without exposure to any of the stressors. The blue one is going to be low dissolved oxygen or hypoxia treatment. Green is low pH or acidification. Red is that low salinity treatment. And then gray is the presence of all 3 of those stressors combined. And so we found that when we exposed 48-hour villager larvae, so that initial swimming stage, to these stressors for 96 hours, we saw reduced growth in response to hypoxia and low salinity. And there's essentially no growth of the larvae when exposed to all 3 of the stressors. Next, when we exposed larvae, the pedovillager larvae, so these are the ones that are ready to settle for 48 hours, we found their settlement, which is the amount of larvae that attached to the substrate that we gave them, was lower, both in response to all the stressors independently or in combination. And then next we exposed spat. So this is larvae that had just settled 24 hours prior for 7 days to these same stressors. And we found that there was increased mortality in the presence of low salinity, which about 75% of the spat died in the low salinity or all three stressor combination. Additionally, these recently settled oysters were smaller in these treatments as well. And then finally, we exposed 4-week-old juveniles. So these that were reared at the hatchery prior to exposure. So these are sort of juvenile oysters that are just were the size of my finger, as shown in that photo. And we found, and we exposed these ones for 24 days in the lab and found that these juvenile oysters, only when they were exposed to all three stressors did you see increased mortality relative to the control, but a lot lower levels. Only about 15% of them died compared to 75% of those day-old spat that died in the all three stressor treatment. And then we also saw that shell growth was slower in the presence of all three of the stressors, in particular low salinity, and shell growth was actually negative when we exposed them to all three stressors. And so this is kind of evidenced by these photos. So on the top here is a picture of one of the juvenile oysters at the end of the exposure and the controlled treatment. So this right here is sort of the new shell growth, whereas in this picture on the bottom is an oyster that was exposed to all three stressors for 24 days. And you can see there's no new shell growth and it's actually sort of rounded out and it is probably dissolving. So the amount of shell they're producing is less than what's being dissolved by the environment that they're in. And then so if we kind of put all these results together and back into the context of the Eastern oyster life cycle and the implications this may have for oyster reef restoration efforts and already existing field populations. So if you deploy culch or have a reef in an area with suboptimal water quality or that's prone to these flooding disturbances, even if you have spawning elsewhere, once larvae enter these conditions, you're going to see reduced growth, which is going to cause them to spend a longer time in the water column, meaning they're going to be more vulnerable to predation, as well as they're going to use up more of their energy reserves, decreasing the likelihood that they'll actually reach the settlement stage, at which point you're going to see reduced settlement under these conditions, which is going to reduce the amount of oysters that you're potentially getting to recruit onto your restored reef or existing reef. And then for the oysters that do actually settle, especially in those first initial days and in the presence of low salinity, you're going to see really high mortality rates, which is going to further reduce the amount of recruitment you're getting to your reef. For the ones that do survive, they're going to have slower growth rates in these conditions, which is delaying the time that till they reach sexual maturity or can become a reproducing adult, as well as reduce their ability to deal with future stressors because of the stress that they were exposed to as juveniles. So really just decreasing the likelihood that you'll have a successful restored reef or that you're going to be able to maintain populations in these conditions. And so this is where the policy brief comes in. So we translated these findings. So this is the first page of the brief. And then on my final slide, I'll actually have a QR code for a link to the project website that has the PDF. But basically, we summarized our main takeaways to that overall flood-associated water quality stressors have negative impact on oysters in their early life stages, which may limit or prevent the recovery of resilience of oyster reefs. And in particular, low salinity had negative effects on all of the early life stages, and newly settled spat were the most vulnerable. So therefore, we suggest salinity as the most important water quality indicator of oyster recruitment at a potential oyster restoration site. But hypoxia and acidification stressors should also be considered because they added to the already damaging effects of low salinity. And in the Mississippi Sound, where this restoration work's taking place, these flood-associated stressors are a real threat. So as evidenced by last year, which was actually the wettest year on record for all three of the Mississippi coastal counties, and reduced salinity levels were observed across the coast during the peak oyster recruitment months of May through September at both water quality stations maintained by MDMR and USGS, as well as sensors we had deployed at St. Stanislaus College in Bay St. Louis. So this graph shows sort of the average salinity and the number of days below 5 or between 5 to 10 parts per thousand. As you can see, 3 of the 5 sites averaged below 6 parts per thousand, which was the concentration we used for our low salinity treatment. And all sites experienced sustained periods of low salinity conditions. Additionally, at the Bay St. Louis site, where we deployed our sensors with the help of St. Stanislaus High School and the amazing marine science program they have, we were able to record pH and dissolved oxygen data during this prolonged low salinity event. And so in these graphs, the horizontal dashed lines represent the conditions in our lab exposures. And so for pH, you can see that the pH levels in the sound were falling between the conditions we exposed oysters to, or often they were below pH levels. And as well, there was a 3-day hypoxic event at the end of July at this site. So this data supports that these stressors do co-occur and present oyster reef habitats, and they may be limiting recruitment success in oyster reef restoration efforts. And so what are the next steps? Because we do believe there is hope for oysters in the Gulf of Mexico, and that the restoration and projects the state is investing in can be successful. But our first recommendation is really incorporating these early life stage tolerance thresholds into existing decision-making tools, such as habitat suitability indices, which are normally based on adult tolerances or just the year-round average of a location, with really salinity being the most important. Additionally, continue investing in the hatchery larval production for remote setting and seed aquaculture, because these are going to be really important restoration activities that will help boost oyster populations in years of poor recruitment due to flooding events. And additionally, sort of leveraging the environmental data sets that we already have. So our team is currently working on synthesizing other embrace-related data and external data sources to really aid in understanding the issues and challenges that affect oyster reef resilience, and then hopefully translating this into another brief that we can share with the community of practice. And then finally, just funding to purchase and install. And most importantly, the hardest part is maintaining these monitoring stations to provide the necessary real-time data from relevant oyster habitats to continue informing oyster restoration and fisheries management. And so with that, I'd like to thank the whole Embrace team that's led by Deb Gotchfeld, as well as all those that helped make these experiments possible, and the interns and Letha Boudreau at St. Stanislaus High School for helping maintain our sensors, and the funding from Embrace, and all the oysters supplied by Auburn Shellfish Lab. And so with that, this is the link to the project website, what I can put in the chat as well, and then there's the QR code. We've got plenty of time for questions.
Yeah, great. Thanks, Jessica. Thanks so much. And I also dropped the link to the project webpage in the chat. So yeah, Jessica, I think questions are going to come in. Because we're recording them and folks won't be able to see that when they're watching the recording, I'll read them for you and then we'll go from there. But so the first question is, so it looks like those 3 stressors might be slightly decoupled based on the graphs from Bay St. Louis. Looks like this, as the salinity is climbing back to normal, that the dissolved oxygen is dropping along with pH increasing. So I'm wondering about the timing of stressors, whether order of exposure might impact survival, or if the stressor occurred sequentially instead of simultaneously.
Yeah, so actually a lot of times with that fresh water, it's going to be more that's coming from riverine sources. It's going to be more acidic than your oceanic water. So when you get these low salinity fluxes, you might also get the low pH then. And a lot of times we see the hypoxic events more in those warmer August, summer months. So you get the additional sort of temperature stressor too. But I think the fact that the low salinity really drove a lot of the responses that we saw, that sort of, even if you sort of change some of the stressor combination, it might actually have a higher impact, but definitely through all the life stages and just in even just a couple of days, that low salinity was sort of driving a lot of the responses. But I think it's definitely important to consider sort of earlier in the sort of spawning season, you might see more of a pH and low salinity. And then as sort of you get, although it might not be as much low salinity sort of towards the hotter months, but then you're going to get into your hypoxic and at a higher temperature, which is more stressful as well.
Great, thanks. Are there others that have questions? I think with this small group, you're welcome to come off of mute and ask your question as well. I know it's a little hard with with webinars to get questions, and so I would just add, as we're waiting for folks to type their questions in, that Jessica mentioned the policy brief. So the policy brief was intended to kind of highlight the key research takeaways of this research for policymakers in Mississippi, and so that we think that is a really useful one if you are just kind of interested in a more general overview, not just of these particular research findings, but in the context of what's been happening in Mississippi related to oysters over, this extended period of time, kind of where we are currently, and certain things that, next steps that might be considered, which, like Jessica mentioned, include thinking about early life stages in existing decision-making tools is one of the recommendations in there. Great. Good. All right. Well, we'll give folks just one more minute to see if anybody has questions or thoughts as well. I also want to thank Embrace for the funding supporting this project, and for giving us this opportunity to look at it. I think this is the kind of second pot of funding the research team has had with respect to Embrace funding, and yes, we're grateful to be able to have that and to share that with everyone. So, okay. Oh, yes, we have a question. Okay. Oh, yes, we have a question, Christina.
I was wondering if oysters are more exposed because there aren't very many of them, and what impact having a healthy population to start off, if any, would make on these results.
Yeah, Jessica, just in case that was a little hard to hear, we had an echo, but I think Christina was saying, like, are the oysters more exposed because there's fewer of them on the reef, and that if there was a healthier population to start with, would you maybe see different outcomes?
Yeah, so I think sort of in years past, we've had sort of these extreme flooding events, but sort of just in the past, this 21st century, there's just not a hardy or a large adult population out there. So it's these early life stages and their survival is really having a bigger effect. Whereas before, you would maybe see it would actually sort of create a clean substrate, then you would get boost in sort of settlement. But now that sort of some of the existing stock quite as high as it was before, it's definitely the earlier life stages, and if they're surviving is even more important.
Okay, great, thanks. I'm starting to see hands come up. I have Ben Posadas next, and then Kevin Felcher. So Ben, do you want to ask?
All right, Jessica, this might be a forward question, but based on the findings that you have and your policy recommendations towards the end, how would you relate that to looking at the effectiveness of the restoration efforts of the oyster reefs past, let's say, 10 years, and those efforts that will be done in the next 10 years, in terms of the type of the materials that have to be planted into the oyster reefs, the timing of the planting or restoration efforts, the density of the restoration materials that have to be put per acre of the reefs, and so on and so forth. Generally, what I'm trying to look at, because I'm also looking at this from a different angle, these results of yours would surely guide some decisions that will be made with regards to restoring the state's public oyster reefs. So what will those be in terms of type of materials, timing of the timing of the planting, and the density of the amount of the coarse materials to be planted?
Yeah, I think sort of some of that sort of substrate question, it doesn't, sort of our work doesn't really get to that, but I think the idea of if you've got culture material or with spat already on it, I think definitely we've seen that just wait between a couple of days to there being a month old, they're definitely a lot more resilient to some of these stressors. So if you're deploying that kind of sort of spat on shell out onto a reef, definitely I mean, it's costly to keep them in a hatchery setting, but sort of as long as you can wait. And if the conditions out there aren't good, it's not even worth the time to put them out there because they're just going to die. But in trying to time sort of deploying your shell as close to the spawning season, because we don't have sort of the science to back this up, but they like clean shell material as well. So, I know just from putting our sensors out there, things just foul up in the matter of weeks out there. So, if you can deploy materials sort of in your April, they might, or sort of towards the May, getting sort of those peak spawns that are occurring in June and July, but also just still getting the data of when sort of these larger spawns are happening is still sort of data that a lot of reefs sort of everywhere. We don't really know when these larvae are in the water. And so to continue to kind of doing our best will require that kind of information as well.
Okay.
Great, thanks. Okay, Kevin is up next. Now I'm on mute. I think you may be on mute. Let me see if I can help. Yeah. There you go.
Good afternoon.
There you go.
Can you hear me?
Yeah.
Hello.
Yes, you're good. Thank you. You're good.
Okay.
Thank you.
Perfect. Thank you. Hey, my question was, are y'all your group currently working on, or have you endorsed any restoration projects along the Gulf Coast in Mississippi yet?
So yeah, we're not directly connected with any restoration projects, but we're sort of part of the restore funding to do science that informs them, and then to work sort of closely with the agencies that are doing those.
Great. Yeah, so our research has been shared with DMR and DEQ, so other folks that are involved in the restoration funding and selection of restoration projects. And so, yeah, we haven't worked directly for particular individual projects, but hopefully it's informing their decisions moving forward. Great. Okay. Thanks. Well, I don't see any other questions, so we'll go ahead and start to wrap up. But if you have one last thought, you're free to put it in the chat. But as I mentioned, we will be sharing this recording of the webinar on the National Sea Grant Law Center's website following today. It may take us a couple days to get it uploaded because we do get captions and transcripts and get that all in order before we post it online. But I will share that information with all of the individuals who registered for the webinar so that you'll have access to that and can share with others in your networks. And so I just want to thank everyone again for joining us today. As if you check in the chat box, you can get a link to the policy paper and also other research papers that our team has done. So thank you for joining us, and we look forward to you coming back to another webinar in the future. And thanks, Jessica, for presenting your research and doing the webinar today. So thank you.
Thank you.